So the next speaker is Margaret Sutherland. Thank you so much for coming. I know this was probably quite stressful. You just walked in during Laura's talk. So you're moving from one place to the next. So Margaret Sutherland joined NINDS in 2007 and serves as the program director in the Neurodegeneration cluster at NINDS, and there she oversees grant portfolios in Huntington's disease, frontotemporal dementia, basic and clinical studies supporting genetics, protein, mitochondrial dynamics, synuclein biology, and stem cell-based research associated with Parkinson's disease. And in that context, she will talk here about how links, neurolinks, standards, and pipelines can be integrated into those neuroscience initiatives. So thank you for coming and then being here with us.
Well, thanks everyone, and my apologies for not being able to make it yesterday. We had council, so I had a couple of grants going through, so the investigators are happy. Anyway, so what I wanted to do today was put a bigger context in terms of what we're doing in the neuroscience space. And I want to demonstrate how you can start from many different places to get to what we can use from the LINKS initiative. And so to start with, I'm just going to mention some of the key neuroscience initiatives that are ongoing now and that I think will play into what's happening in LINKS and NeuroLinks specifically. So one example is the NINDS Parkinson's Biomarkers program that started in 2012. We enrolled 1,500 people, collected biosamples, distributed data, and that program led into what is now a new initiative for the Accelerating Medicine Partnership in Parkinson's disease. I'm going to mention the NIH Common Fund Extracellular RNA Initiative, how that plays into what we're doing in neuroscience, the Microphysiological Systems Tissue Chip Program, LINKS, NeuroLinks, and Answer-ALS, and also give you an example of, again, how natural history studies and what we're doing in them will contribute cell sources that can really be used in a LINKS type program. So this is my perspective in terms of how we see human platforms, and we're definitely, my focus is definitely in trying to understand human biology and use sources that take us to a better understanding of the cells underlying that biology. So you can start from many different places, as you can see on this diagram. You could start with the biobanks. You could isolate brain cells either through induced pluripotent stem cell differentiation into different cell types. Or lately, through the Brain Initiative, there's been a big focus on actually doing single-cell transcriptomics and hopefully in the near future, proteomics analysis. So again, we're going to have a very rich set of data to look at. And all of these platforms, in many ways, can be cross-cutting because, as I mentioned, whether you start from a tissue, whether you start from a derived pluripotent or induced pluripotent stem cell, you're still looking at platforms that go across those types of analyses. So you're looking at RNA-seq, you're looking at proteomics, you're looking at metabolomics. And that's why we think that what LINKS has done, especially on the metadata side and potentially on some of the pipelines, can help drive what we're interested in neuroscience. So for us, how we look at things is basically, we definitely want to pull the clinical data together with omics platforms. And we think that clinical data is definitely a key component and a driver in terms of understanding systems biology for neuroscience. And you can see examples of where we would get clinical data from, the omic platforms we're thinking of, and then the types of data that get generated. So to lead into one of the examples, the NINDS Parkinson's Biomarkers program, like I mentioned, started in 2012. Basically, the idea was to standardize, and I think this gets into a metadata structure too, where we wanted to basically standardize the data collection, so no matter what study came into the program, there will always be a core set of clinical assessments that we did. So when you look at a study, you can pull up core assessments and go across studies to really start developing an understanding of what the disease is, regardless of the state of the individual. And so this biomarker program basically also relies very heavily on what we call the DMR, which is our data management resource. And you can see that from a researcher level, basically we have an electronic data entry system that brings the clinical data into the data management resource. We have a built-in data dictionary. We have form structures built both for clinical data, and now we're building out for data that comes from biomarker analysis. We've tied the data management resource with the biorepository so that you can actually query clinical data to find the types of biospecimens you want. And all of this infrastructure basically is what's contributing or going to add into the Accelerating Medicine Partnership in Parkinson's disease. This is the underlying module system that we have for the data management resource. Basically, Proforms is the electronic data entry system. The GUID is the global unique identifier. We use it to basically create encrypted ID that can track a person across studies. So let's say an individual, a patient has enrolled in this biomarker study, but they also may want to be in a clinical trial. We'll be able to link that data through this GUID at the individual level to learn more about that individual as we go forward. So it really ties into what we see as a potential tool for a more precision medicine-like approach to the diseases we're interested in. The data dictionary is a key thing for us, and it helps everyone coming into the data management resource understand how the data was created, what the variables are, and as helps them consider how they should do the analysis of the data. The query tool is really where you find all of the studies that you're interested in. And right now it does a 3-way link, but we hope in the near future we'll do a 5-way link across different form structures. And so because we've built it so that the metadata is shared across all studies, once every night basically, we've built basically queries that look and build aggregate data on the public site so people can get a look and feel in terms of what's available in the data management resource before they actually log in. I was going to show you some form structures, but in this case I'm going to show you how we can then move from a biomarker-driven study into something in terms of building out cell resources. So that global unique identifier basically helps us to link across clinical forms. So in this case, what I've done is I looked for family history of Parkinson's. I looked for the age at diagnosis. The upset is a smell test that you can get a total score on. And then the NDS-UPDRS is basically the standard that the Movement Disorder Society uses to measure Parkinson's clinical symptoms. And so we've also, through Parkinson's, developed a genetic rating scale. So again, you can take the values from the genetic rating scale and identify extremes in that scale and look for individuals that fit within that context. And that's how we're building out our cell source for this Parkinson's biomarker program. The other thing we're doing is helping the tissue chip program that's run through NCATS. It's in its second stage. The first stage actually started out as an NIH Common Fund project, moved to NCATS. It was focused on toxicology initially. The second phase is focused on disease modeling. In this case, a grant that was funded focuses on Parkinson's and ALS. So again, we want the cell sources to come from our biomarker program. So we looked in this case for early onset Parkinson's cases, and we're developing cell sources based on that. The tissue chip program is interesting in that we're right now in the LINKS program, we're really doing 2D disease modeling. In the tissue chip program, they're trying to build a 3D disease model of either ALS or Parkinson's disease. And this gives you an example of the chip that's being used for Parkinson's and ALS. It comes from Emulate. It was actually used in the first phase of tissue chip. And it basically creates 2 channels, one where you'll be able to introduce astrocytes, microglia, neurons, and on the underside you build a blood-brain barrier. So in this way, having that 3D environment enables you not only to look at the cells and how they function, but also to look at metabolites and other things that are released from the cells. You can collect those, do proteomics, metabolomics, etc. on that type of system. And we think by having a more complex system, we'll be able to model disease more accurately because in the brain, it's not just made up of neurons, it's actually a very complex architecture. This gives you an example. This is from Kai Swenson's lab. I don't know if I can, maybe I can, yeah. Where he started with ALS, and actually the cell source comes from Neuralinks. So the iPSC lines that were developed by Neuralinks are actually being used on this tissue chip right now. The concept is that, again, you're going to be able to introduce multiple cell types onto the upper chamber, and then look at how the blood-brain barrier impacts function underneath. And this next slide gives you an idea of how, from a tissue chip, will also be able to link what we know about human biology through doing comparable assays in the biomarker setting. So you can see at the bottom there's the omics platform where they're going to look at proteome transcriptomics. SWAP is one of the proteomics approaches being used by Neurological right now, as well as RNA-seq. There's also, the chip also allows high content imaging to be done, and from the effluent you can do other types of analyses to get an idea of what's happening to the cell, as well as with the tissue chip, you can do live cell imaging, so you can get an idea of network activity using multi-molecular related approaches. So that gives an idea of how you can start at a biomarker type of program. You identify the cells you want to work with for the patient, and then you can start using very complex types of in vitro systems to start doing disease modeling. For the NIH Accelerated Medicine Partnership, that started in 2014. It's a partnership between NIH institutes and industry. It's a 50-50 partnership, so 50% of the funding comes from industry and 50% comes from NIH. Francis Collins was the initiator of this partnership. It started out with type 2 diabetes, Alzheimer's, and rheumatoid arthritis, and this week we announced that Parkinson's was going to be added to the matrix. And I think, again, it emphasizes that many different approaches are being used. For the type 2 diabetes, most of the data is genetic data, and it's based on a growth platform. For the Alzheimer's disease, all of the data so far is based on RNA-seq from tissue. For the rheumatoid arthritis, lupus is actually single-cell RNA-seq from biopsies. And then for the Parkinson's platform, which I'll show you, Verily is one of our partners. They're a Google Alphabet company. Stefan Zuckner might have mentioned that Verily is also involved in the All of Us program, and so part of what All of Us is going to develop as a platform will be brought into our Parkinson's Accelerated Medicine platform. So this gives you an idea of what the partnership looks like, but again, also it gives you an idea of what the data is going to be. So we already have existing data for about 4,000 whole genomes. Right now, in the first stage of the program for Parkinson's, we're going to be generating 7,700 whole transcriptomes. And stage 3 is going to break out and look at metabolomics, proteomics, and decisions are made based on the robustness of the platform and reproducibility. So this type of data comes from biosample analysis, but you can see how by developing cell sources, you could also go in and ask in your cell model, can you actually come up with similar data sets? So, and again, this just gives them a breakdown in terms of why we're able to pull it together, and it's really because there are existing efforts, not just by NINDS, but also by the Michael J. Fox Foundation and the Harvard Biomarker study. So these groups came together, they collected samples in a similar way, which makes it easy to do the biomarker analysis. And you can see that there's, especially for CSF, there's quite a bit of CSF available that's a longitudinal sample set for us to look at. And as I said, as we move into stage 2, we're going to be looking at proteomics, both unbiased and targeted, and probably focus on CSF and plasma. And again, I think hopefully what we are doing with proteomics will learn from links. Because one of the challenges, depending on the platform, is how you capture that data and how you make that raw data available. So I think the whole genome is pretty standardized. It's something that the community recognizes as a certain way to capture that data and work with it. RNA-seq is becoming that way, but proteomics and metabolomics are still major challenges in terms of standardization. So I wanted to mention the Extracellular Common Fund Initiative because, again, since we're interested in signatures for people with neurological disorders, the Extracellular RNA Common Fund has done some really interesting things in using CSF and plasma to look at what the extracellular RNA species are in those biosamples. And one of the interesting things, unlike the whole transcriptome, where you're going to get a lot of messenger RNA, in the extracellular, in the CSF and plasma, we tend to get more of the regulatory RNAs, and that's what we're very interested in The initial data comes from Kendall Jenner's lab, Jensen's lab, sorry, and it does demonstrate that I think there's enough, at least looking at the entire CSF plasma, there's enough reproducibility that we can consider using that as a platform in the accelerated medicine partnership. This just gives you an example again, like Lynx has done, they took the approach of developing metadata standards where they too can pull and give the investigators an idea of what's in the database before they actually go in. And I think that's a really important point to always have a landing page where people can initially get an idea of what's available so it intrigues them to go further in terms of pulling or working with data sets individually. I mentioned the PD knowledge platform before. Verily is going to be the builder behind it. It's all going to be cloud-based. And I think, at least calling in yesterday, sort of a few things that it's a different type of approach I think that investigators are used to. What we want to encourage is investigators to keep the data in the cloud. If they want to bring in their, or work on their data sets, or bring their data sets to the cloud, and also the pipelines they're interested in. So we want to support cloud usage. We want that money or the investigators' effort to really go into optimizing how they manage and handle data. And it also, I think, will increase the sharing across projects in that. So it'll be an experiment. We're going to obviously pull together user groups to get feedback. But I think when we build funding announcements, the funding announcements will be supporting that type of approach to keep it in the cloud and avoid handling. So I talked about Parkinson's. Another thing that I cover is from temporal degeneration. I mentioned this in case anyone's interested in funding. The National Alzheimer's Project Act is a law that was established back in, I think it was about 2012. It has, over the last few years, added about $400 million a year to the NIA budget. Part of that money is directed towards Alzheimer's-related dementias, and temporal degeneration happens to fit into that. So it's an unusual place to be. If you're an investigator interested in Alzheimer's or Alzheimer's-related dementia, there are funding announcements and pay lines that don't exist for other diseases. So I would suggest that you find out more in terms of what opportunities, if you're interested in this space, are available. For us, we funded 2 natural history studies. One, especially is focused on longitudinal evaluation of family FTD. In that case, we've actually recruited prodromal individuals who are gene positive for one of the genes that cause FTD that have not shown clinical symptoms yet. Then individuals who have manifest FTD, and then the ones in that family who are gene negative. And so we think it's going to give us a handle on terms of actually looking for biomarkers that will tell us about the prodromal state and how change occurs into manifest FTD. And so major genes involved in temporal degeneration include progranulin tau and the C9orf72, which is an overlap with ALS. So the ALS FTD phenotype definitely have overlaps in terms of the genes involved. I should mention too, at the bottom you can see our investment besides collecting the data. And I wanted to mention this is one of the problems I'm seeing right now. The clinical data resides at the National Alzheimer's Coordinating Center. The biospecimen data is at NICAD, which is their DNA repository, and the imaging data resides in Lonnie. So unless you're a very educated investigator and know a lot about these studies, it might be obvious how you find the data you want to do analysis on. And we've also begun to develop self-sources for these natural history studies. Again, something that could potentially pull into a links type approach to analysis. So through ADRD funding, one of the first funding announcements that came up supported Center Without Walls on tau pathophysiology. And to help the centers, we developed cell sources from those natural history studies I just mentioned, where they're carrying different mutations in the tau gene. And one of the centers funded actually focuses on functional genomics. And it's an interesting approach in that initially they started out using siRNA to understand how different genes impact phenotype. They then built libraries where they had combinations of 2 siRNAs so they could look at whether the impact of 2 genes was complementary or if it had basically a neutral impact. And that they built data and libraries that can start looking at other types of phenotypes. So one of the things in FTD we're interested in is how the tau protein is transmitted between cells, because in humans you can actually see a spread of that protein when you look at autopsy tissue. So one concept would be if you could stop that spread, then potentially you could have an impact on the disease. So this is one way they've done it. And you can see what I mentioned is that when you do that two-gene combination, you can either see it's a buffering effect or a synergism. They started with siRNA, but now they've moved into a CRISPR, basically repression/activator system, which again gives them a lot of capabilities, but it also creates lots of data sets. And these data sets are cell-based, and again, something that we could think about if we're going to expand on the LINKS program, where we're not just looking at a perturbate that's a ligand, for instance, but we want to understand how genetic perturbates could actually impact phenotype. Another component of the Center Without Walls is run out of Giovanni Coppola's lab, and they've actually developed and looked at ALS or site RNA-seq data. And again, this is a public site you can go to put in RNA-seq data. In this case, I typed in LINK2, and what happens is you can see at least an initial analysis of what the gene expression looks like. In this case, it's hippocampus and striatum. So what we like to see is ways to take these types of visualizations and data sets and pull them together to work in a LINKS type of environment. So Neuroscience, as you may have heard of already, basically was brought in at the second phase of the LINKS program. And it supports understanding some of the neural cells that are present in the brain. And the concept of Neuralinks is to focus on transcriptomics, proteomics, epigenomics, and imaging. And in this case, a single center basically differentiates the cell types, and then they send either cell pellets or live cells out, differentiated cells out to the different groups who are doing these platforms. The data comes back in, and I think what makes LINKS and Neuralinks important right now is we really are interested in how to integrate these types of platforms. So we want a signature not based just on RNA-seq or whole genome sequencing or proteomics. We want to understand what those data sets mean in terms of a complete signature. So I think that's the challenge with Neuralinks, is also how to take that cell data and relate it and give it human relevance. So you're starting out with human cells, but you want to know the pathways and targets that you're identifying are very relevant to what you're seeing in human disease. And I think we can develop ways of actually going back and looking at data sets, for instance, derived from tissue sources versus what you're actually seeing in a cell-based environment. I think the other thing about Neuralinks that we're really interested in is how to have sustainability. So this program has a life, as many programs do, but the data that's been generated actually is valuable because we feel that the cells that the groups have been using are actually useful for other groups to ask different questions about. And so NINDS has developed or has invested in making the cell lines from Neuralinks available to our cell repository. But we also want to make sure that the data flows on too, and some of the tools used to look at the data are available as well. So this was a concept that started maybe a year ago, and Ajay's been a driver behind it. And we're definitely interested in working with LINKS and Neuralinks to figure out how to not just sustain, I would say, how can we actually build it out to apply to other disease areas. The other challenge I see with LINKS, and it's like I showed you with our natural history studies of Untitled Chapter, some of the solutions for handling the data live in different environments. And if you're not an educated user, if you're a naive user that isn't familiar with the program, it's very hard to know where to go to get the data and then how to pull it all together. And so what I've heard from postdocs, even in the LINKS program, is that they would like to see a platform where you just go to one environment and everything's there, and you have the tools to work with the data. So we're hoping that we can find a solution like that going forward. And I think the Accelerated Medicine Partnership in Parkinson's disease has that concept. We want a single environment to bring in all those data types with the tools to help people do the analysis.
And so one of the questions that I have, and we're dealing with it in our Parkinson's biomarker program, is how do you work with raw data proteomics when potentially groups are using different platforms? And proteomics and metabolomics seem to be heavily driven by some analyses that are done by the equipment itself, so you don't necessarily get the raw data set out, you get a somewhat processed data set to work with. And there are also file formats that are potentially proprietary, so how do you visualize those types of data sets? And then, like I said, is how to integrate that output in a common platform. And finally, and again, it's in one way, as I mentioned, we started with natural history studies, Parkinson's, raw temperature generation, did the biomarkers, worked into cell sources. In this case, in ALS, it basically grew out of the concept that Neuralink seems like a good idea for working with cell sources. So they put a lot of investment in to developing iPSC lines from every individual that's enrolled in this. I would still call it a natural history study, but it's a concept that could lead to a precision medicine approach. And so 1,000 individuals is the target for enrollment. IPS lines are developed. And then again, ALS has that same problem where clinical data resides at Mass General. The whole genome sequence has been done at New York Genome Center. Some of the links, or Neuralinks, as you know, some of it resides at MIT and Ernest Frankel's lab for epigenomics, transcriptomics. But there's not a good integration yet of all those different data types and how you develop that signature, whether it's from a biosample perspective or from a cell source perspective. So I think those are the challenges I see. I think there's possibilities here for helping us solve some of those. And I hope I give you the impression that it's not just about Neuralinks, but I think in a lot of projects and initiatives, large ones happening in neuroscience can actually benefit from a platform that brings everything together, provides tools and visualizations that will help different levels of users. So there'll be the data scientists who just want the data, but there'll also be biologists and other types of investigators who want something that's a little more processed and easier to handle. And I think for us, especially on the accelerated medicine partnership side, we want to make sure we address everybody's needs. And it'll be a challenge, but I think it's doable as long as we bring user groups together like you and get input in terms of what you're looking for and what you're doing. And with that, I'll close. Any questions?